Welcome everybody to the Precision Nanosystems Tea Time webinar series. For those of you who've been to this series before, welcome back. And for those of you who are new, and this is your first one, thank you for joining, and I hope you find this session informative and educational. So today we have Dr. Kirsty Maclean from Codex. Kirsty will be talking about accelerating biotherapeutics, utilizing synthetic biology strategies. So before we get started, I'd just like to mention that our next Tea Time webinar series after this one will be on August the 26th, and we'll have Dr. Lawrence Paul from Curadigm, and she'll be discussing using a nanoprimer to improve delivery systems of various therapeutic agents. So Kirsty, over to you.
Fantastic. Well, thank you everybody for joining this afternoon, this morning, whatever time zone you're in. And as AJ mentioned, for our little chat today, and to be quite honest, I could really kill for a good cup of tea myself right now. I've been in the US for several years, and it's kind of bereft of some good tea bags. But that aside, so for our talk today, it's going to be biotherapeutics accelerated. And what I mean by that is kind of discussing, as AJ mentioned, kind of methods of synthetic biology. So we're going to talk about automated methods for this, for your fragment builds, your library builds, as well as your clones using our BioXP system. So I think this word sustainability is sometimes an overused word, but really what's driving the need for some of these renewable resources and in fact better therapeutics and alternative approaches to agriculture is actually our increasing population growth. And that's especially true over the last 50 years. And that looks like it's going to continue to do so, as you can see with this graph. Now, fortunately, synthetic biology approaches actually affords an engineering approach to biology to address this problem. And we can use tools to design and build functions in cells as a means to implement actually these improved agricultural, food, biofuel, as well as therapeutic processes. Now, we as humans have actually exploited microbes for thousands of years. And what I mean by that is we can supply bacteria and yeast, for example, with simple sugars. And I'm sure you all know we can produce goods such as bread, wine, beer, and cheese. And as our ability to actually now reliably and rapidly engineer organisms improves, these living cells will increasingly be used to produce goods and therapeutics at industrial scales. Now, historically, really Genentech kind of inspired all this. So historically in 1976, they actually developed a synthetic version of human insulin using at the time what was called a new gene editing technology in bacteria. But that actually demonstrated for the first time that it was possible to produce human insulin and actually other therapeutics in bacteria. Now, when we think about advances in synthetic biology and actually where we are today, it's really had quite a profound effect and impact actually in oncology. Now, specifically when one thinks about rational design of immunotherapies based on bioengineered cells, I think some of these efforts have been well described around T-cell therapies, especially CAR-T and TCR, and I highlight aspects of that actually in the cartoon. Now, CAR-T, and I'm sure you've had seminars on this from others, so I'm not going to go into it in some detail, but CAR-T is obviously known as the living drug, and it's obviously received a lot of attention scientifically as a promising anti-cancer approach. And I was just reading last night about one of the first patients, Doug Olson. He's in his 70s now. He was actually the third patient 10 years ago this week to receive novel CAR-T treatment for CLL, and actually he remains in remission to this day. So obviously very effective for hematological malignancies, not so much for solid tumors, which we'll get into. So briefly what you see in the cartoon with regard to CAR-T and really kind of strategically thinking about synthetic biology approach, briefly kind of autologous or allogenetically derived T cells are genetically engineered to express a certain chimeric antigen receptor, which then programs a series of events to target specific antigens that are present on the tumor surface so that they can recognize and destroy the cancer cell. Now, again, very successful in hematological malignancies, not so much for solid tumors. So to that end, a lot of strategies have been developed around the TCR. So while CAR T cells actually recognize the protein expressed on the cell surface, the T cell receptor, or TCRs, can recognize tumor-specific proteins on the inside of cells. Now, specifically TCRs, and you'll see again from that cartoon figure on the lower right, they actually use heterodimers that consist of alpha and beta chains. And this is going to come up actually in my talk in the next 10 minutes. And these actually recognize fragments presented by MHC molecules. So T cell therapy certainly affords, I think, a much broader range of targets compared to CAR T. And as I mentioned, it's been a very effective approach for solid tumors. But there are several disadvantages, and in fact, well-described disadvantages to this approach that I'll allude to here. So really, as a reminder, aspects of T cell therapy are MHC restricted. And what I mean by that is their efficacy actually depends on presentation by that MHC molecule to recognize targets, demonstrate an optimal binding affinity, and therefore activate T cell function. So, of course, obstacles include that binding affinity to tumor antigens, the selection of good targets, the search for specific TCRs, and of course, screening for optimal TCR affinity, and thinking about other indicators such as the safety evaluation, for example, toxicity, and of course, the time and cost involved in all of that. But another important consideration is actually the immunosuppressive microenvironment of tumors, and really how the orchestration of immune cells, fibroblasts, endothelial cells ultimately influence T cell trafficking and function. Now, of course, that's enough for its own talk outside of even today, but suffice to say, there's actually been a lot of advancement in the design and construction of novel drugs affecting this. And as such, you've got the design now of these checkpoint inhibitors, and as well as TCRs that actually improve T cell trafficking and activation. Now, despite these advantages, we're still seeing late stage clinical failures of drugs, and of course the need to develop personalized and tumor specific approaches to cancer. So with that, I kind of propose a provocative question. So how can a synthetic biology approach reliably engineer more effective, importantly, precise T cell therapies? So with that, I do want to quickly summarize who we are as a company and what we do to build biology. So this story really started in 2008 when Dan Gibson, and that's who he is on the right-hand side there, Dan Gibson, who's actually our Chief Technology Officer, while he was at the Craig Venter Institute doing his postdoc, he recreated the genome of the Mycoplasma genitalium bacterium, as well as creating the first living cell with an entirely artificial chromosome. Now, these seminal papers obviously heralded that synthetic biology era, but also heralded in a novel cloning method, which I'm sure you've all heard of. It's the Gibson assembly method, which has been used today in well over 5,000 publications, and obviously has many patents associated with it. But it's the Gibson assembly method that's actually fundamental to our technology. So briefly in 2013, we were founded as SGI-DNA, which was actually a spin out of the Synthetic Genomics Institute. Then in March of last year, we spun that out as SGI-DNA. We actually got a Series A financing complete late last year. And as of this year, in April 2020, we have rebranded to CodexDNA. So what I'm going to do for the next kind of 5 minutes is actually introduce our system. It's actually a very exciting time in our company. We're just about to launch our new system, which is the BioXP 3250 system. And really, it's for scientists needing that simple, fast, secure method for nuanced cloning needs, especially associated with updating your T-cell receptor. And this is really an ideal solution for some of those workflows. So I'm just going to go through aspects of that again over the course of the next kind of couple of slides. So again, briefly, this platform can really revolutionize your DNA synthesis needs, especially to improve immunotherapy and personalized medicine approaches. So it makes synthetic genes in an automated manner. We can accommodate up to 32 reactions at any given run, so 32 fragments, clones, or libraries. Now, at least in the US, it's a consistent 3 to 5 days from placing that order on our secure portal to then achieving that final product. Again, that final product could be your fragment, your clone, or your library. So it's really a combination of a hybrid of insourcing and outsourcing for your synthesized DNA needs. Now, with regard to immunotherapy and of course personalized medicine, we've certainly touched on various applications associated with T-cell therapy, specifically thinking about CRISPR applications. Obviously, the design of suicide switches, that's been a very novel approach now, especially to deal with the toxicity of engineered T-cells. Also, the design of bispecific T-cell engagers, which are engineered antibodies consisting of 2 single-chain variable fragments with affinity both for that CD3 as well as the tumor antigen. But also some of these engineered tumor antigen approaches, especially neoantigens, and aspects of that I'm actually going to be discussing in today's case study. So again, we've got many publications touching on all these areas of biology, and this is an example of the system on the right hand side. So again, what is the system? Again, just to kind of summarize, it will consolidate all your workflows. So you're really building your DNA at the bench with all our kits. Again, we package everything for you, so it's simply about loading up that instrument, which takes about 5 minutes, and then walking away. So it's fully automated, push button. Overnight, you can generate, again, your fragments, clones, or library builds. Of course, versatile, you can deploy it now in kind of any research or clinical settings. It has its IQ, OQ, and PQ validation. As well as driving productivity, and aspects of that I'll allude to in the case studies that kind of discuss several of our customers' experiences, especially around thinking about novel T-cell therapies. But importantly, it's IP protection. Again, with an understanding of how Gibson assembly works, we just need to know the homologous ends. So we don't need to know the makeup of your backbone vector, and that vector should never leave your own lab. So it allows you and affords you the flexibility, again, to design some of these more nuanced constructs, again, and you have full control over that. So I'm going to spend, again, the last part of this talk kind of walking you through a couple of case studies, which really kind of integrates this particular system into T-cell therapy workflows. The first of which is actually a recent paper that came out in Nature earlier this year. It's actually from Gigammune in San Francisco here in the US. And they certainly allude to an issue at hand, much of which I've actually discussed over the course of my summaries, and certainly allude to some of the issues with the T-cell receptor. But the goal of the paper was actually to employ and integrate some of these high throughput methodologies, and some of you are, I'm sure, very much aware of some of these methodologies, but integration of all of these to really identify clinically relevant TCRs with high functional avidity from diverse T-cell repertoires. And this is an example. I pulled this exactly from the methodology as described in the publication itself, so I'm not kind of massaging it in any way. But just to kind of walk you through that, you can see it cartoon represented on the left, and I've kind of alluded to the workflow here on the right-hand side. So the group actually used droplet microfluidics to isolate millions of individual T cells into some of these what's called droplet emulsions. And then they physically link those to the TCR alpha and beta chains from the single cells. Now, these TCR alpha and beta chains and were created to make a library, and that was actually cloned en masse using the BioXP into full-length lentiviral constructs. And again, I allude to some of the aspects that was accommodated on our system, the BioXP in front of you. Now, these lentiviral constructs were then transduced into GERCAT T cells, and again, you're creating that TCR alpha beta GERCAT library. Then, of course, you can highlight the process again with the subsequent validation and kind of mining for sequences, and subsequently the group were able to mine for TCR alpha and beta sequences using peptide MHC panning, for example, as well as TCR mediated activation screens, and then of course deep TCR sequencing efforts. So again, a lot of different technologies integrated into that entire workflow. So really this kind of repeated screening and kind of panning approach enabled by the construction of some of these libraries actually identified more than 2 million, in fact almost 3 million natively paired TCR alpha and beta clonotypes. So achieving a much more efficient throughput approach, again, integration of a lot of these high throughput technologies. Now, I think these type of efforts kind of really lend themselves to thinking about integration of a lot of technologies into one's workflow. And really to that end, I think it does remove some of these scientific bottlenecks that until recently have existed, especially for TCR discovery. And in this case, thinking about the development of new adoptive cell therapies. So I'm more than happy to share with you. You can certainly put it into the chat, or if you want to email me, I'm more than happy to share with you a link to that specific paper. Now, for the last part of the talk, in a similar approach, I want to spend the next 5 minutes to focus on some recent work actually presented by Steve Schoenberger of the La Jolla Institute of Immunology in San Diego, and actually work stemming from his new company, which is TCRx Oncology. So Steve's lab is actually focused on understanding the T-cell response following infection or even following tumor development, and to that end, he's been focused on identifying these neoantigens expressed in both kind of mouse models and human tumors, and he's actually optimizing several methods to target them. Now, I'm going to kind of walk you through this kind of very specific process. So with the recent approach, several of his colleagues have actually now developed this very kind of robust as well as scalable process for rapid identification and validation of these tumor neoantigens. So if I get your attention to the top right, it all actually starts with genomic sequencing efforts to identify tumors with the shared driver mutations. This is then followed by a proprietary assay to identify the patients with specific mutations in that T cell. And then you have single cell genomics to obtain the mutant-specific TCR, and then you're generating cell lines to express the mutation. And then this is followed up with use of the BioXP to identify candidate TCRs. And finally, you're treating the patients with these engineered approaches and these engineered T cells that are specifically targeted to that driver mutation. Now, I've kind of got a walkthrough process of that in steps 1 to 8, which essentially walks through exactly what I've just stated. But I do want to illustrate that this process actually takes time. Unfortunately, you can't get away from that, the time associated with that in steps kind of 1 to 3. Again, it's all about the sequencing, and a lot of that is, you know, through use of Foundation One or other companies. But certainly steps kind of 4 to 7, that is a highly time-consuming approach, especially at the bench. It's a highly iterative process. And as was alluded in the kind of GigaMune paper, this is a huge bottleneck in kind of moving that needle forward for T-cell therapies. And in fact, in some cases, it can take up to months just to identify a single TCR candidate. So really, the group certainly wanted to adopt this BioXP system to allow them to dramatically decrease the time to identify some of their TCR vaccine candidates. So I wanted to take you through a couple of slides and kind of walk you through an approach with a specific patient that actually had pancreatic ductal adenocarcinoma, and this remains actually a devastating tumor with poor prognosis. So this particular patient, as you can see kind of in the bullet points, obviously had this SOP for this tumor type, surgical resection, both neoadjuvant as well as adjuvant chemotherapy, also checkpoint inhibition, and the use of kind of nucleoside analogues. Unfortunately, as you can see from the imaging, disease certainly progressed, and there's associated ascites as well as liver metastases. So there's clearly a need for a more novel approach. So again, thinking about that workflow that I alluded to, now in the arena of genomics, of course, we can look for some of these druggable targets. So with the use of sequencing, we're actually able to identify a specific mutation in KRAS. It actually causes the KRAS G12V driver mutation. This seems to be quite prevalent in pancreatic tumors, also actually in lung tumors. So really the question is, can we find specific TCRs against these driver mutations? So again, thinking about step 3, again, this is using the proprietary T-cell assay, again, developed by Steve Schoenberger's lab. Perhaps we can identify patients with mutation-specific responses. And here you see a number of peptides representing the kind of common recurring cancer mutations. Of course, you've got the KRAS mutations represented, as well as BRAF, and of course, I'm not sure if you can see on the x-axis there also kind of PI3 kinase mutations. Now, from an ELISpot, the patient did actually have a response to the specific KRAS mutation. You can see that highlighted, as well as the SMAD mutation, and that's actually a common mutation occurring in pancreatic cancers also. So obviously, we've got some very specific responses. So now we actually want to be able to detect that response from T cells in the peripheral blood. And so what the group did was to perform, again, single cell sequencing on these T cells, again, to elicit a response and identify the relevant TCR alpha and beta chains. So at this point now, we can enter in the integration of the BioXP system. So we can seamlessly clone each of these candidate TCRs now into a lentiviral backbone. And I highlight in the middle, actually, the makeup of that backbone construct. And obviously, you can't read it. That's probably a good thing, although this is now publicly available information. And then on the right-hand side, I'll kind of walk you through the approach whereby we use the BioXP automation to perform Gibson assembly, again, to identify some of these candidate TCRs. So again, you've got Gibson assembly of candidate TCRs into our proprietary lentiviral backbone. Test this Gibson assembly again using the BioXP constructs in these J76 cells. That's a Jurkat cell line. And then move into transduction of these into primary T cells with the TCR candidate, and then test the candidate for tumor-specific recognition, both in vitro and in vivo. Now, the last few steps, and I've kind of consolidated a lot of work here, really kind of it does involve a lot of the verification and validation steps that you would expect, when working with some of these patient samples. So of course, you've got cell surface expression of candidate TCRs and verifying that that is true, also verifying the antigen specificity. And one also has to confirm the recognition of naturally processed antigens and of course verify that patient TCR actually recognizes the appropriate HLA restriction. So again, all told, after all this work, and of course that workflow that I kind of alluded to, this slide kind of really summarizes the goal of that very strategic approach, again incorporating that novel T-cell assay, as well as sequencing, single cell genomics, and obviously the use of the BioXP. So I'm not going to go through every single one of these specific bullet points, and of course the final product is an off-the-shelf TCR immunotherapy, proven to naturally recognize some of these driver mutations, and in this case the KRAS G12 mutation. This of course could be relevant even, for example, in lung tumors as well, not necessarily restricted to pancreatic tumors. Again, all of this is within the context of a restricted MHC. So certainly I think this is really nice work. Some of it's already been published. We do actually have a YouTube video that walks you through some more granular detail as to this approach, and I'm more than happy to share that with anybody that's interested. So again, just to kind of summarize really the approaches of really why was the BioXP used, and really what are the advantages when one thinks about personalized medicine. Well, really I want you to focus on the two top bullet points presented here. It's really about the fast turnaround time over conventional methods, especially benchtop cloning methods, which I think Steve's lab and that of GigImmune alluded to with the kind of tedious workflows. But it's all about the efficient, high quality builds that you'll generate in just an overnight run. And of course, the instrumentation incorporates a very novel two-step error correction process that is unique to the instrumentation outside of any benchtop approaches. So of course, we do afford a very robust application development, of course, a wide arena of genomic workflows, again, some of which I've alluded to in the talk today. And again, I'm more than happy to share with you any publications that touch upon these relevant areas scientifically, especially in personalized medicine. So again, just to summarize really how we kind of impact some of that scientific research. Again, I've alluded to areas within personalized medicine, especially around IO, and really thinking about improving the validation cycle times. Again, it's all automated. Again, you can use your own proprietary vector and protecting your own IP. And just again, some of the quotes from Steve Schoenberger in that regard. But it also lends itself very nicely to, of course, biologic drug discovery. Again, decreasing the time to results, especially when one thinks about the design of DNA libraries. And again, improving kind of lead optimization time, you know, really in the case of 6 to 50 times. And of course, we've been very instrumental in some of our vaccine development strategies, of course, around tools for the SARS-CoV-2. And we were able to build that entire genome in just over a week, really from design all the way through execution to that 30 kb genome size. And that includes now some of the mutations associated with that. So again, I thank you for your time. I'm more than happy to take any questions this afternoon, this morning, and of course you can feel free to email me at any point if you want some more detail or examples of publications. But again, I thank the guys at Precision Nano for hosting today. Again, think about Precision Nano for some of your downstream needs with aspects to what I've presented today. Again, it's all about integrating many of these workflows really to better science going forward. So thanks, guys.
Thank you very much, Kirsty. That was really interesting, very, very exciting field. Lots of potential, so really interesting. So that neatly brings us to the top of the hour. So all that remains is to say thank you once again, Kirsty, for a very interesting and thought-provoking presentation. Really appreciated it. Thank you for everyone for logging in today. We hope you found that interesting too. And hopefully we'll be able to see you in 2 weeks' time for our next Precision Nanosystems Tea Time webinar. Thank you, everyone.
Thanks, guys.